Hello and welcome back to Psychology 303, the analysis of everyday behavior. I'm Dr. Williams, and this lecture will be the first over Chapter 8. Here we go. Chapter 8, Respondent Conditioning. Okay, so what exactly is a respondent? It's a fancy word for a reflex. Reflexes are very simple behaviors. Up until now, we've been talking about voluntary behaviors, behaviors that we choose to engage in, behaviors that are complex in nature, that use skeletal muscles rather than smooth muscles, and behaviors that respond to contingencies of reinforcement or contingencies of punishment. But a respondent is a reflex, and it's governed by the reflex arc. They're biologically involuntary, and they usually consist of very simple responses. Let me give you some examples here. Respondents are controlled by the central nervous system through the reflex arc. I'll show you a diagram of that in a minute. And respondents are stimulus specific. So reflexes don't just happen to any stimulus. They're usually it's a very specific stimulus that causes a reflex or a respondent to occur. Typically, they occur without any training at all. They're very, very basic. They're often present from birth, or they may emerge at sexual maturity. And they include things like suckling and salivation and gagging and coughing, eye blinking, startling, choking, shivering. Your heart beats is a reflex. It increases or decreases in response to certain stimuli. The fight or flight responses are reflex-based responses. The reflex function, the function of how a reflex works, was first described by René Descartes in the 1600s, and he's the one who coined the term reflex. That the energy from the environment was reflected back through the nervous system. So he's the first person to really try to explain how reflexes work. These are some of his classic diagrams that demonstrate the biological connection of the sensory organs like our skin, and the pathway to the brain, and then the pathway back out to respond. So here's a little boy who is too close to the fire, and so he feels the heat. It goes up through these, and these were considered, these veins or ducts went up to the pineal gland, where it turned around and came back out as a response. And they thought that it was hydraulic, that it was dictated by the flow of essences or humors that cause these reactions. Today we know better. This is a cross-section of the spine, and it diagrams the three neurons that are associated with the reflex arc. We have receptors in our skin that detect sensation, and there are neurons called the afferent neurons that go from the receptors in the skin through. You can follow the little arrows here. I'm going to use my cursor. So here's the receptors in the skin. We feel something on our skin, and the receptors pick up that sensation. They send that information through all the way to the spinal cord. And at the spinal cord, inside the spinal cord, there is a special neuron called an interneuron that works as sort of a U-turn for this information. And what happens is that the information just simply turns around and stimulates the efferent neuron, which goes out to the effector muscles, which causes us to respond. So when you touch heat, the information comes in, it spins around and goes right back out to your muscles to pull your hand away from the heat. Now there are also neurons that go from here to the brain. So if this is at the level of where your arms are, for example, it would come in from if you put your hand on a hot stove, your sensors would pick it up, it would send it to the spinal cord, and it would whip around and go back out, and you would pull your hand away. It would also send messages to your brain, where you would process it cognitively and go, ouch, with language. But the actual response by the effector muscle requires no cerebral involvement, no brain involvement. Years ago, literally hundreds of years ago, Galvani, a very famous scientist in Italy, did an experiment where, this is back like 300 years ago, where he stimulated the neurons in a decerebate frog. In other words, he only had frog legs. He didn't have the whole body, and it didn't have a brain. And he would stimulate the efferent neuron and cause the leg to jump. It would pull back. And so they were able to demonstrate that these neurons, the afferent neuron, the interneuron, and the efferent neuron, could operate independent of the brain. So we get the information when we use our reflexes and we pull our hand away because something's sharp or something's hot. We respond, and we know that we respond, but often the behavior occurs before our awareness, before we actually are aware that we've done it. Okay. So the primary work on respondent behavior happened in the 1800s with a Russian researcher Russian physiologist named Ivan Pavlov. And he was actually studying digestion. He was interested in the salivary gland, and he wanted to see if the salivary gland could be conditioned to work more or less or sooner or later. And he used dogs primarily because they were convenient. At the time, there were lots of stray dogs on the streets of St. Petersburg. He rescued these dogs off of the streets. He brought them into the lab and gave them a home. And while he was doing his salivation research, he accidentally discovered what he called classical conditioning. So I think it's best if we review some vocabulary before we continue. Most of you have been exposed to this vocabulary before in Psych 101, but I think it's a good idea for us to review it. And so there are several important components to classical conditioning. The first two are the natural reflex itself, which consists of a stimulus and a response to that stimulus. And that stimulus and its correlated response are naturally occurring. They're not learned. They're part of our biology. And so Pavlov referred to these as the unconditioned stimulus and the unconditioned response, or the unconditional stimulus and the unconditional response. And what he was basically referring to was that they were unlearned. These were the stimulus response patterns that are selected for in our biology. We come with them prepared. There's no learning involved. But classical conditioning is about learning to elicit a reflex with a different stimulus, one that's not biologically predetermined. So initially we call that stimulus a neutral stimulus because it does not cause the reflex. The neutral stimulus, or NS, is a stimulus that does not cause the reflex that we're studying. It doesn't mean that it doesn't do anything. I mean, if it's a bell, for example, a dog might turn and look at it. They might respond to it, but they don't, for Pavlov's dogs, they don't salivate to it. So it doesn't produce the reflex that you're studying. And through a series of pairings, we end up with a new relationship. An association is formed. And what we end up with is that previously neutral stimulus becomes a conditioned stimulus after we've gone through our conditioning or training trials. And now, although it did not previously produce the reflex we were studying, now it does produce the reflex that we're interested in. And once that reflex is being elicited by a novel stimulus, we now talk about it as a learned response or a conditioned response. The word conditioned means learned. So the same reflex, but now it's being produced by a conditioned stimulus that previously had no effect on the behavior, and now it does. And that's what provides us with evidence that learning has occurred. Okay, so let's talk about the basics of classical conditioning. This slide is a little bit on the small side, but it's a summary of what you've probably learned before and what's covered in the book. So there are 3 stages to classical conditioning. There's a series of tests that we do before we start any conditioning. So prior to conditioning, all the stimuli have to be tested to make sure that they're doing what we think they should be doing. So first we have to select a neutral stimulus, and then we have to test to make sure that it doesn't produce the reflex that we're interested in studying. We then also want to make sure that we have the correct reflex and the stimulus that goes with it. So our unconditioned stimulus, we need to test it and see if it produces the unconditioned response. So in Pavlov's example, he used dogs and he used a ticking metronome. Kind of a ticking sound, tick, 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 tick. And when he observed the dogs and he put the ticking mechanism in the room, he noticed that the dogs did not salivate. He did not get an unconditioned response. That's exactly what he wants. He wants the metronome because it doesn't produce salivation naturally. What does produce salivation is food powder. So he tested the food powder on the dog's tongue, which is our unconditioned stimulus, and it does make the dog salivate, which is our unconditioned response. So prior to conditioning, we're simply testing to make sure that our neutral stimulus does not produce the reflex and that we do have a correct stimulus response reflexive pattern with a stimulus and a behavior that we control. Once we have those, we can then begin our conditioning trials, which consist of what's referred to as a pairing procedure. Now the neutral stimulus is paired together with presentations of the unconditioned stimulus. So the NS is added to the US, they're presented together, and we get a UR, we get an unconditioned response because the US is being presented. So this is repeated over many, many trials. So in our example, Pavlov used a ticking metronome, and he would let the metronome go tick, 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 and then he would present food powder to the dogs. And then, of course, the dogs would salivate because the food powder makes them salivate. But now they're getting the food powder in the presence of that metronome. Tick, 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 food, tick, 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 food, tick, 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 food. And each time the food makes them salivate. After many trials, we then can conduct a testing procedure to determine whether or not conditioning has occurred. If the neutral stimulus is presented alone, so now we present just the NS, and we look to see whether or not the reflexive response or the reflex happens to just the presence of the NS, just the neutral stimulus. If it does, we can say that the neutral stimulus is no longer neutral, and it now becomes a conditioned stimulus. We call it a conditioned stimulus. We have evidence to show that it's no longer neutral and that learning has occurred. So the reflex now, tick, 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 the dog salivates, tick, 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 the dog salivates. We can now say that the ticking metronome is no longer neutral, it's a conditioned stimulus, and the salivation is no longer natural, it's learned with respect to the metronome, and so we call it a conditioned response. So it's when it's emitted to the sound of the ticking instead of the food powder, it's now a conditioned response. And this is what Pavlov discovered. Here's a diagram of what I just described. So we have food, which is our unconditioned stimulus, and we have salivation, which is our unconditioned response. In 2, we now, before conditioning, you can see that there's a bell, and the bell does not produce food. In slide 3, we have the bell paired with the food, bell food, bell food, bell food on many occasions. And the dog salivates because the food is present. But with enough conditioning trials, with enough of these pairing procedures, after conditioning, we can now ring the bell and see the dog begins to salivate. And we now no longer call the bell neutral. We now call it a conditioned stimulus. And the salivation we now call a conditioned response. So that's the basics of Pavlov's experiment. At this point, I'd like you to pause the PowerPoint and go to the assignments lectures video file and watch Chapter 8B, Pavlov's Respondent Conditioning, for a little video that has an overview of these discoveries that he made. It's a little simulation. So why don't you go do that and then come back and we'll continue our lecture. Okay, welcome back. The couple things, so now that you have an understanding of what classical conditioning is all about, or respondent conditioning, it's called Pavlovian conditioning, classical conditioning, or respondent conditioning, or associative learning. They're all the same thing. You'll hear different phrases, different books will use different terms, but they're all referring to the same reflexive conditioning. So let's talk a little bit about the relationship between the neutral stimulus or the conditioned stimulus and the unconditioned stimulus, and how the timing of the presentation of the NS and the US together can affect conditioning. So on the right, I have a diagram. Let me show you how to read this. I'm going to take my cursor and put it over here. So we have forward delayed conditioning here, and we have a diagram. And so what we have here is time traveling across the bottom here. And these little diagrams are classic examples of how we diagram classical conditioning. And they deal primarily with a line that shows when the CS is off, and it comes up and it stays high when the stimulus is on, and then it goes back down when it's off. So this basically describes when the conditioned stimulus is off, when it's on, and when it goes off again. And the same is true for the US, when the US is off, when it's on, and when it's off. And so now we can look in terms of time when each of these stimuli goes on and goes off. So with forward delay conditioning, we see that the CS comes on shortly before the US is presented, and they overlap. Okay, so the neutral stimulus, or what will become the conditioned stimulus, comes on before and then overlaps with the unconditioned stimulus. With trace conditioning, you'll notice that the CS comes on and goes off completely before the US is presented. So it comes on and is terminated before the US is ever presented. That's called trace conditioning. Simultaneous conditioning, they come on and go off at the same time. And backward conditioning, the US, now in this case, the US comes on first here before the CS comes on and goes off. And they may overlap or they may not, but the more they overlap, the more it will help with learning. All right. So higher order conditioning is another phenomenon associated with respondent conditioning. And here's the deal. The key to good respondent conditioning is predictability. When a neutral stimulus reliably predicts the US, when it tells you that the US is coming, and it does so in a very reliable way, we form an association between those two stimuli, and we learn to rely on the conditioned stimulus to anticipate the unconditioned stimulus. And when we do that, we learn about it and our behavior becomes conditioned. Well-established conditioned stimuli, so once you have a really well-established conditioned stimulus, you can use it to condition other stimuli. So we can use a CS that we've already learned about to condition another stimulus. And these links can be extended out to more and more stimuli. So here's my example. I'm going to use my cursor again here. Here is our natural reflex right here, the US-UR relationship. That's our biological reflex, the unconditioned stimulus and the unconditioned response. Once we pair CS1 with the US, we now have formed an association between these two stimuli, and with enough training, CS1 will produce the response, now a conditioned response. It will now produce a conditioned response. And once it's really well trained, I can then pair another stimulus, a CS2, to CS1, leave out the US, and still get a conditioned response. And then I can use CS2 to train CS3, and CS3 will ultimately produce the conditioned response. Let me show you a picture of this. So here is a diagram of higher order conditioning. And this is just 2 levels, so it just goes to second order conditioning. So the food and the salivation, here the food is the US to produce salivation. And he adds a ticking metronome. So here's our neutral stimulus. It becomes CS1 through Pavlovian conditioning. And now the metronome alone will make the dog salivate. So now we can take the metronome and pair a black square to the metronome. And with enough training trials, the black square will produce salivation. Couple of little things you should know. One, there is a limit to how many levels. Here, I'm going to go back. There's a limit to how many levels, how far back you can get. Could you get a CS4? Could you get a CS5? And it really depends, literally, on the strength of the unconditioned stimulus. How strong that stimulus is determines how many levels you can go back. If it's not a very strong stimulus, you may only get 1 or 2 levels back. But if you have a very powerful stimulus, like a stimulus that produces a great deal of fear, you might be able to get quite far back. Okay? Food is a very powerful stimulus. And so you can train food back several steps. That's why they don't just call it second order conditioning, it's really called higher order conditioning. All right. So I'll end with a little cartoon. Here's the little dog in Pavlov's device where he did his work. And the little dog is talking to another little dog and says, "Watch what I can make Pavlov do. As soon as I drool, he'll smile and write in his little book." All right, we'll see you back for lecture 8B.